Hello, my name is George.
Hi, my name is Lahiru.
And today we're going to be talking about a frameshift mutation and how it is caused. Frameshift mutation is this genetic mutation that occurs, which is a random change in the DNA sequence. Frameshift mutation is also known as a reading frame error, and this reading frame error is caused by an insertion or a deletion of an amino acid. We look at the genetic code in terms of triplets, which are going to cause a change in the bases of the amino acid in the sequence of our DNA. All in all, this is going to give us a weird or funky frame that is not the same as it would be without this mutation. What we get is a new translated protein relative to the normal, meaning it's not as functional or there might be something missing. There is dependence on how early this mutation occurs, which will overall give us a means of understanding if it's going to happen more often or what other changes will happen in the future. Some of the impacts of frameshift mutation is on the length of the protein. What happens is that we can get a stop codon. These are going to cut or stop the sequence from continuing. We get an increase or decrease of the length of the amino acid, as we talked about, caused by a stop codon, and how these can be non-functional.
So an example for frameshift mutation would be Tay-Sachs disease. It's a genetic mutation caused by the degeneration of nerve cells in the brain and the spinal cord. So the gene that causes Tay-Sachs disease is called HEXA gene, and then it's located on chromosome 15, position 23 and 24. So with patients with Tay-Sachs disease, in 1990s they reported that they were showing resistance to infection, especially HIV retrovirus. How to figure out if there is a frameshift mutation? We look at translated mRNA codes, and then we look at the letters in groups of 3 bases, basically codons. And then we focus on one amino acid group at a time. Then we use amino acid group chart.
This chart is the chart that kids who are trying to figure out what each one of these triplet codons refer to this chart. It's broken down into all of the possible outcomes that you can get, as well as the different variations that will give you that same type of amino acid. Let's begin with our very first example, which is about deletion. Deletion is the act of having one of your amino acids removed and the frameshift moves to fill in that spot. Let's look at it in terms of the DNA, which is our normal. What this deletion mutation does is that the C is completely removed, bringing the whole frame to be shifted to the left.
Another example for frameshift mutation would be insertion. In a normal DNA strand, we have CAG as the first codon, CCC as the second codon, and ACT as the third codon, with the respective amino acid chain. When an insertion occurs, which causes a mutation in frameshift mutation, it causes an addition of a T nucleotide into the second codon, which causes pushing over of all the other nucleotides into the third codon.
A better way to understand this will be to look at it in terms of general terms. It's easier to picture what's going on if we were to look at it in terms of the normal, giving us a sentence or a phrase of "see the dog run." However, when we have a frameshift mutation, this phrase is altered. So a deletion alternation of the S letter in the phrase will mean that we get something weird, like this. So S is gone and now we get EET instead.
And then for insertion, what happens is, for the normal one, see the dog run, where at the second position we have addition of the E, which causes the pushing over of all the other amino acids into the third, fourth, and the fifth respective.
Thank you for watching this video. I hope you understand a little more about what a frameshift mutation is and how it occurs.